Recorded. Uh, if you've been following this series, you know that we post uh, our programs to the Native Orchid Conference website, and we've just begun posting them to YouTube. So if you are coming in late to this program or would like to see it, watch it a second time, or refer a friend to it, you'll be able to do that fairly shortly. A couple other housekeeping notes. Please make sure that your microphone is muted so that we don't interrupt the speaker. There will be time for questions at the end of the program, and there is a chat option, as I think most of you know. With that, I'd like to introduce our speaker. Jordan Teisher is Vice President of the Biodiversity Research and Director of the Herbarium at the Missouri Botanical Garden. At the present time, he is co-leading the Revolutionizing Species Identification Project, which, if you read the MailChimp that went out earlier or have been following activities in the herbarium world, this is a multi-million-dollar project to digitize the botanical garden's nearly 8 million specimens, many of them orchids, as you know, and develop new tools for plant identification using artificial intelligence and spectroscopy. So this is a glimpse into the future, I think, and it's now my pleasure to turn the program over to Dr. Teisher.
Hi, thanks so much. Can you hear me okay?
Just fine.
Perfect, thank you. Thank you so much for having me. It's a real pleasure to be here virtually with you talking about this project. Um, I wasn't quite sure of the background of the group, so I thought I'd start out first just introducing what an herbarium is, in case there's anybody in the group that is not intimately familiar. So an herbarium, when we talk about them, is a collection of preserved plants, usually dried and pressed. There are terms that were called herbaria early in the history of the world that were more illustrated books like herbals. But over time, Luca Ghini, an Italian medical doctor in the 16th century, had the innovation to press plants between the pages of a book and dry them in order to allow his students to study plants in the winter. So at that time, the herbarium was also called Hortus Siccus, the dry garden, or Hortus Hyemalis, the winter garden. Uh, as you may be aware, a lot of medicines were based on plants at the time, and so this enabled a much better year-long study of the medicinal plants compared to the drawings at that time that were sometimes a little bit rudimentary and hard to make out the exact details. A pressed plant, if you've ever pressed and preserved flowers or some other plant, maintains all of those features that you need to identify it beautifully preserved. There were a series of innovations that I won't go through in the history of herbaria, but a big one was with Linnaeus, the father of modern classification, who had the insight to break those pages apart out of the book. That way they could be moved around according to classification changes. So as you rethought the classification or as you re-identified a species, you could keep them close to other specimens that you thought were the same thing and near one another in the cabinets. And this is the way that large and small herbaria are mostly organized today. Here's a picture from Kew Herbarium in London. So inside each of these cabinets is a separate sheet like this. I don't know. Can you actually see my cursor? I guess I should have checked on my mouse cursor circling this thing.
Yes, we can.
Okay, perfect. Each one of these sheets pulled out separately and then stacked inside the cabinets of the herbarium. Uh, they are still used for the original purpose for which they were created by Luca Ghini, which is for education. They're a tool in universities around the world to teach botany to students. But I'm also going to talk a little bit later about some of the new applications of herbarium specimens. So our own herbarium here at Missouri Botanical Garden, I'm currently in my office a little bit after hours on the garden grounds. Our garden was founded by Henry Shaw, a wealthy businessman in St. Louis in 1859. It was open to the public. Slightly before that, he sent his friend George Engelmann, who was a botanist and doctor in St. Louis, to Europe to purchase herbarium specimens and library books. And the goal was to establish the Missouri Botanical Garden as both a botanical garden for the enjoyment of the citizens of St. Louis, as well as a research hub, as St. Louis was at that time still the gateway to the West. Western North America was, compared to Eastern North America, relatively poorly described botanically. And it was thought that having a research botanic garden at this gateway to the West would be a tremendous asset to global botany. So Engelmann went to Europe, purchased a private collection from Johann Jakob Bernardi of about 62,000 plant specimens, which formed the nucleus that expanded to become the herbarium today. It started out in this museum building, which is still on the garden grounds, modeled after the museum building number 2, I think it's called, for economic botany at Kew. This is an inside view of what it looked like at the time. Outgrew that after about 50 years. They expanded our administration building to accommodate the herbarium, lived there for another about 60 years. And then today, it's split between 2 different buildings. So in 1972, the collection moved into the Lehman building, which is where I'm sitting right now. That's on the garden grounds, if you've ever been to Missouri Botanical Garden. And then as it outgrew that in the mid-90s, they constructed a second building, the Bayer Center, and moved half of the collection there. So we have a few logistical obstacles that some collections don't, being split between those 2 buildings, but it's a very large collection. As Jan said, almost 8 million specimens. The herbarium grows by about 40 to 50,000 specimens per year in normal growth. That excludes large acquisitions of collections that are donated in bulk. So private collections that are very large or university collections that are closing and are donated to us. The average, if you include those over the last 10 years, is about 100,000 specimens per year. We're somewhere just shy of 8 million. We have 7,156,417 as of January 1st accession specimens. So those are ones that we have an actual count for. Every time that we mount a specimen, it has an accession number that counts 1 up on the sheet. And then we have about 800,000 unaccessioned specimens, still accessible, still part of the collection, but they haven't gotten that accession number stamp yet. So we're a little bit, if you're familiar with other herbaria, a bit unique in that regard for large collections of having that accession numbering system. About 4 million of those, a little over 4 million, are digitized in some capacity. So they have usually basic data associated with the specimen is on our public website called Tropicos, tropicos.org. Right now, you need a login for tropicos.org. If you haven't checked it out before, the login is free. We're just getting scraped so hard by bots that it keeps on crashing the site. So if you go on there and you get an error message saying you need to log in, that's why, but you can set up an account and still access the database. It's still intended to be public. We just have to find new ways of dealing with bot activity. We have a little over 3 million images. These numbers were just updated recently. And right now, as part of this RSI project, I'll talk about a little bit later, we image about 25,000 specimens per week. So the grant is to do a total of 6 million additional images and 4 million transcriptions to get the full herbarium online. So this process is tremendously labor intensive. So if you think about those 8 million specimens and where they came from, this is botanists going into the field. There we have Mary Morello on one of her expeditions a few years ago at this point, pressing plants that she's collected between pages of newspaper, putting them into a plant press. There's Roy Giroux, another one of our curators, carrying a full heavy plant press in the field. They are shipped in boxes back to an herbarium. They have labels prepared for them after they're dried. They typically pass through the freezers. And then plant mounters glue, stitch, and tape those specimens onto cotton paper with the labels. That way the specimen is protected a little bit against that sturdy cotton background, and it has the label associated with it forever. So that is a lengthy process, as I said, multiplied by 8 million, and you have a bit of a sense of the amount of human effort that has gone into assembling a collection like this. The good news is that if you take care of them and you keep them away from bugs and fire and mold, as far as we can tell, they last indefinitely. So that specimen on the left is our oldest one that we have found in the collection. It's from 1692. In the middle is a specimen from Captain Cook's first voyage, and on the right there is a specimen collected by Charles Darwin on his voyage of the Beagle. So these are some of the historic specimens in our collection. This one they all traveled the seas in boats and in trunks, so maybe a little bit beat up, but all of the details are there that you would need to identify those specimens, and they're still useful scientific specimens, 300 years later. So one of the main uses is we are still completing that basic inventory of the plants of the world. About 380,000 accepted species. I haven't updated that recently. It might be slightly higher. I haven't looked at World Flora Online's total. But we think there's something like 10 to 20% still remaining to be described scientifically. There's about 2,000 described per year, give or take the year. We have about 200 species described here at Missouri Botanical Gardens. This is a major center for new species descriptions and documentation and naming. And we are describing those species slower than they are often being lost from the wild. Also, because of the nature of how you find species, the ones we're finding now tend to be rare. So there was an estimate that came out not long ago from, I think, some people at Kew that something like 70% of the remaining species would be expected to be rare and of conservation concern. So it really feels like a race against time. I'll return to that theme a little bit later about describing species and putting names on them. So another way that herbarium specimens are useful is in reconstructing the tree of life. So you can, in most cases, get usable DNA out of herbarium specimens. The DNA tends to be a little bit degraded, so you have to do techniques that lend themselves to applications with degraded DNA. But for example, in a recent Tree of Life project, about 28% of the samples for that project, which is a massive sequencing project, came from herbarium specimens. You can get some of these specimens benefit from what we call ancient DNA techniques. So you extract them in a sterile environment, like you would if you were trying to extract DNA from a mammoth bone, for example, that you found in the ice, and you're worried about some kind of contamination. That can help improve your quality. You can also get DNA from specimens that are unavailable in the wild, and so you might not be able to find them again, or it might be cost prohibitive or geopolitically complicated to get them. As in, a war zone, we don't typically give DNA without the permission of the country where the specimen was collected. But this is a great way also of building that, what we call a phylogeny, that tree of life. And you can do all kinds of things with phylogeny in terms of evolutionary analyses. But one that you can do is this conservation of evolutionary diversity. So you can ask yourself, well, in this great suite of species that we have, which should we be the most concerned about? And one way that you can look at this is, well, maybe we should conserve the ones that are really, really evolutionary distinct. So that means they are on a branch of the evolutionary tree kind of all by themselves, and they don't have a lot of close relatives. The assumption being that those will have a lot more genetic differences than closely related things. So that evolutionary diverse and globally endangered edge species is an application where you can use this evolutionary relatedness as a proxy for, or as a guide for conservation purposes. That evolutionary relatedness also has predictive value. So if we're thinking about useful plants in medicine, in foods, for providing genetic diversity for some of our crop species, knowing how plants are related to one another gives you insights potentially about what species could help substitute for a lost species ecologically, could be a place that you would want to look for medicines, for useful chemicals, etc. As we digitize a lot of specimens, they can be used in some complicated modeling, especially as computers have gotten more sophisticated. So as you start to build up thousands and thousands or hundreds of thousands of records of the same species, you can do interesting things like, this is a bit of an older study now, but it's one that I like for this ice plant, Carpobrotus edulis. And it's South African in origin. And what you can do is map all of the herbarium records that you have. So those are all these little red points on this map onto the world map, and then overlay climate data that you have for the world. And then you extract from each point where you have an herbarium specimen or specimen record, the climate variables at that location. And you can build then a climate profile or kind of a climate footprint for that particular species that says, all right, here are the conditions that this species seems to like to occupy in the world. And then in this case, Carpobordus edulis is quite an invasive species in several different parts of the world. So you can use that to construct a map of where this species would like to live. And maybe some of those areas are areas where it hasn't been found yet. So for example, you see this, the darker the color blue, the more suitable the habitat is. A few years ago, I was visiting my sister in California on the coast, and my nephew was delighted with, he was about 2 at the time, delighted with all the ice plant that had invaded the California coasts. You can do the same thing, and that helps you set conservation or invasive management priorities of who should be looking for this plant where it isn't there yet. Or conversely, let's say you have a species of conservation concern, you can do the same kind of climate modeling to say, where could we try to relocate the species if its original habitat is gone? What climatic habitats might it be amenable to? Um, One distinction that we can make here is you see I have a few different databases that have numbers of specimen records. It's a little bit outdated for Tropicos and probably the others. But there are primary databases where you're directly putting records into, like Tropicos. And there are aggregator databases like iDigBio and GBIF. And they both serve important purposes. But I'll talk a little bit later about these primary databases in the context of this RSI project, where a lot of those updates are happening and the names are being actively worked on. So another application, and the last one that I'll go over, is we can capture through new technologies data that is ecologically interesting and relevant from herbarium specimens. So we can use them to extract things like leaf shape. So this is Leaf Machine, a program developed by Will Weaver, who is a postdoc right now, I believe, at the University of Michigan. He was just visiting. I should really know which university he's with. I think it's Michigan. Uh, and this program extracts the leaf outlines from herbarium specimens, converts them into these silhouettes, and then measures the total area of the leaves, for example. And it can do this, churning through thousands and thousands of specimen images using computer models. So what would once take somebody, months and months to measure by hand can be done while you go do something else. The computer's running on the images, but it does require the specimens to be imaged, and so it can access them. We can also scan. So this is X-ray fluorescence using X-rays onto the specimen, and you can use that to detect the presence of heavy metals. So this has applications for ecological remediation of contaminated sites where you have, let's say, industry left heavy metals in the soils, figuring out which plants can tolerate those heavy metals, which are drawing them actively up into their tissues. It can also be used in a process called phytomining, where certain heavy metals like nickel that are now in high demand because of our electric car batteries, they're very difficult to mine. They're intensive to mine and expensive out of the ground. Nickel exists in some soils in fairly high concentrations. So if you can find nickel hyperaccumulators who are actively drawing the nickel out of the soil, you can then extract the nickel from the plant tissue. And that's hence the phytomining. You're sort of mining heavy metals out of plants. And herbarium specimens can be used with these portable X-ray scanners to identify species that are candidates for that kind of a process. So it's already kind of Star Trek-y, and certainly not why they were initially collected in the 1500s to make herbarium specimens. But every so often as technologies develop, we find new and interesting ways to use herbarium specimens to study ecology, to study evolutionary biology, study genetics. And this is one of the really common questions we get as we're digitizing and giving tours of the herbarium is, what are you going to do with the physical specimens after you scan everything? And the answer is we're going to keep them forever and ever as long as we are able, because a lot of these applications, the DNA extractions, the scanning with X-ray fluorescence, those are things that you can only do with the physical specimens. And they could not possibly be anticipated by people collecting those specimens 100, 150 years ago. So all of those techniques rely on species identifications. So we have to have the herbarium specimens accessible digitally, and we also have to have them identified as species. So if I go back just a little bit, when you start downloading records like this on GBIF, each of those records comes from an herbarium specimen, and someone had to put a name on that specimen. And that name had to be, is really a hypothesis about evolutionary relationships that someone or some group of people had to come up with to apply to that group of plants. So these names, it's kind of easy when you go to something like GBIF and there's 110 million records, you can pull up all of the names on there to feel like the plants kind of come pre-labeled. And if you walk around a botanical garden, it feels like the plants come pre-labeled, but it's always important to remember that they don't come that way. If you go out into the wilderness, into the woods, you just are faced with the sometimes bewildering array of natural diversity. And so we are, again, in that race against time to try to name all the species that we can find before they're destroyed. And to do that, we need access to high quality specimen data, ideally available anywhere. And so that's how this revolutionizing species identification project came about. It recognized three kind of key challenges. One is the lack of availability of our specimen data at Missouri Botanical Garden digitally. We only had, less than half the collection digitized in any capacity, and less than a quarter of the collection was imaged, and it had taken us about 20 years to get there. So our first goal was to complete digitization of the herbarium. Second is we need new tools to speed up the identification process. We have some phenomenal taxonomists at Missouri Botanic Garden who are incredible to watch identify specimens, but there's just more plants than any small group of people can really hope to identify. So we need new tools to speed that process up. And then third is we're also not seeing as many taxonomists, so we don't have as many taxonomic training programs, at least in North America and in Europe. And so a third component to this was, we need the data that we're building those models off of to be the highest quality that we can. Otherwise, the models that we build are not going to be useful. And we wanted to build future capacity in taxonomy by training a new generation of young botanists in this curatorial mentorship program. So those are the three components of the project, and I'll talk about each of them a little bit separately. First, I want to acknowledge this is a big endeavor. This is only part of the team. The project is funded through the generosity of an anonymous donor. It's about $14.4 million over the course of 6 years. It hires 21 additional staff members. So these are just the two scanning teams. So on the left, and this is Matthew Austin, who's my co-PI on the project. This is the hyperspectral scanning team, and I'll talk a little bit about what we mean by that a little later. And this is our visual scanning team doing the basic digitization, split between our two herbarium buildings, 5 and 5, each with a supervisor. Not pictured here, which are a couple key players in the development of the project, were Patty Arnold, and Dr. Gunther Fischer, who some of you may know as an orchid person, who were the primary contacts with the donor, getting this project going and developing the overall theme of the project. So first, when we talk about digitization, this is Jetty again, one of our supervisors on one of our imaging rigs. This is a two-part process. Well, it's multi-part, but two main parts. Taking the image and converting the label data that's on that image, I don't know if you see it here, I'll show you a picture in a minute, into a searchable format of standardized fields on a database. And by basic data, what we mean are the who, what, when, where, of the plant collection. So here we have a specimen of Tillandsia anceps, and it says, when it was collected, who the collector was, where it was collected, any notes that would help you describe the plant, ideally some geographic coordinates so it can be mapped, and then accessory metadata that help other people locate these collections. And as I mentioned, one of the things you can do with these data once they're all digitally available is you can map them to help you understand species distributions. So this can be useful for conservation purposes, for identifying species, identifying differences between species. Uh, but it's not automatic getting there. So there are errors. So this is the same set of data. Here's the data from Tropicos. They've been cleaned a little bit. The same species, if you search on GBIF, you might see a couple points kind of in suspicious locations where there are probably not specimens. This is, by the way, not in any way to shame GBIF. You know, as an aggregator, they're handling many, many, many more records than we have on Tropicos. And if you look at natural history data, this is the norm. There's going to be a lot of messiness. It's very hard to convert all of those data for even 8 million specimens into accurate coordinates. And the key thing to remember here is that, when you have errors like this, there are all kinds of clever analytical tools that you can use to clean those data automatically. So I could run a script through these data and say, give me every specimen record that is in the ocean that's not on land. And it can pull all those back and it will flag these records for me and say, here are two that you should probably look at. They're not, these are land plants and they are not growing on land. You can also ask, is the coordinate mapped to the country that the specimen says? So for example, in this record, if I mapped out this coordinate and it's a different country than Panama, and it's supposed to be in Panama according to this location, you can design your database that it flags that record and says, hey, I think this is an error. You should probably look at it. What is much harder to flag and know if it's an error is that species name. And this is a real challenge because when you look at this and you have them all mapped out like this, it's very hard to tell which is wrong. You know, let's say 15% of these are wrongly identified. If this is one of your wrongly identified specimens, that dramatically changes your inference about what the geographic range is of this species. You know, if it's this one, then basically your range is more or less unchanged, although this might be in an interesting habitat that would tell you something about the ecology of the plant, of that species. But for example, if it's this one, or let's say it's these four are misidentified, well, now for your conservation purposes, does the species grow in Ecuador or does it not? It's critical that we have accurate names on these specimens, and it's the hardest thing to verify in any kind of an automated way. So again, that's where this revolutionizing species identification, which is, you know, an aspirational name, kind of a lofty title, but that's our goal, is this process needs to be revolutionized, especially because these data are being used all the time. So we have, you know, over 800,000 downloads from GBIF of our Tropicos shares data with GBIF. So if we just look at the Tropicos data that we're providing to GBIF, it's been downloaded over 800,000 times. There are more than 3,000 citations of studies using these data points. So we really, really need them to be as accurate as possible. That species name is the link for all of the other information that we have about that group of plants. And so it's critical that the name be accurate and that it be consistent and findable. The specimen digitization process is also critical in building what's been called the extended specimen network. So the idea here is that you have those original field collections out of which you make physical specimens, and you take images of those specimens and you enter them into a database, and that gives you the digital record. So you have a digital record and your physical specimen. But you also might have taken a bunch of other things, either at the time of collection or afterwards. So you can take field photographs to complement your specimens. Colors don't often preserve well on herbarium specimens, so it's really helpful to have pictures of the flowers. The context is sometimes lost and is difficult to describe in sentences of how the plant is growing, what its habit looks like, where it's growing exactly. So again, those are helpful additions. You can take tissue samples that you store in a separate collection for DNA work. You can extract morphological data either from the specimen before it's pressed or after it's pressed, store those into a database. The DNA extracts themselves, the DNA data that comes from those extracts. And those all can be linked to specimens. And the real power there is that let's say I go through and I do a bunch of morphological measurements of a particular tree species in the tropics, and I'm interested in how, let's say, elevation affects leaf area in that species. And I go and I spend months and months and months in the field collecting all this data, and then I publish my results. And then we find out that the thing I was calling one species is actually two species. What happens? If I don't have a way of confirming which of my original plants was each of those species, I now have a very confusing data set. These are measurements from two different species that may have very different patterns in the character that I was looking at. Versus if you collect specimens as vouchers for those plants that you're measuring, and you change the names on the specimens, because of this connected digital extended specimen network, you can always extract those data and get them linked to the correct names. So this is why vouchers, these physical specimens that represent the species in the wild, or the specimens in the wild, are the gold standard for this kind of work, because they can always be confirmed, they can always be updated, and serve all of these, new applications in the future for conservation assessments and distribution mapping and all the applications I talked about earlier. So that's the basic data component of digitization and what we hope to do with it. The next part is using new tools to identify species, since it's a difficult process and it takes a long time, and we're always fighting against time. So one tool that we're using are hyperspectral data in a spectroradiometer. So hyperspectral data is the full range percent reflectance. So if you shine a light onto an object, it produces to our eyes a color back at you. Jan, it looks like your top is red. So it's red because it is absorbing non-red wavelengths of light, and it is reflecting red wavelengths of light. Chemically, that's the reaction that it has with those wavelengths. Plants are green because of chlorophyll absorbing wavelengths of light that are not green. But there are many more wavelengths of light that you can use that are outside the visible range. So in this range from about 350 to 2500 nanometers, that's the measurement used for wavelengths. About this range is the visible spectrum. And then outside of this, there's the near infrared and then the shortwave infrared. So we are dramatically increasing the information that we're measuring with the spectroradiometer compared to what you can see or what can be measured just in the visible spectrum. So we have a device, I'll show you here. The spectroradiometer has a probe that you place onto the leaf against a black background, because black absorbs all of the light, so it's not going to reflect anything back at you. And then we shine a beam of powerful light onto the leaf, and then the device measures in a very, very sensitive way what the reflectance at different wavelengths along that spectrum. And what you get is a curve that looks like this. And that curve appears to be specific to particular species. So it's as if species have kind of a spectral fingerprint that corresponds to that particular species, and it's different from the fingerprint from other species. This work got started in Brazilian herbaria about 10 years ago. They started using infrared scans for this. So we're not the first group to do this, and we're not the only ones doing this right now. It's a big international initiative. There's a collaboration called iHerbSpec, the International Herbarium Spectroscopy Group, led by Janine Cavender-Bears at Harvard University. I think it's 11 different countries that are currently involved. But the idea here is to try to build a library of these hyperspectral scans that are consistent and transferable. So I'll talk a little bit about what I mean by that in a minute. Another thing that you can get out of these scans is because different compounds and elements have different reflectance patterns, you can also extract trait data from the scans. So, different wavelengths correspond to the nitrogen content, for example, or the leaf mass area, the amount of lignin in the leaves, the cellulose content, etc. So you can do a lot of interesting ecological things. It's kind of like if you've seen Star Trek and the tricorder. that they have where they scan something and say you're healthy or you're not healthy. It's a little bit like that. It's kind of sci-fi-y. So when they're scanning this, there are a lot of considerations that you have to do. It doesn't quite happen as easily as a tricorder. Presumably the tricorder in Star Trek universe went through a series of testing periods and standardization periods while they worked all the kinks out. That's the period that we're in, or the phase we're in right now, is we don't quite know what is going to matter and what is not going to matter. There's a few things we know do matter. So for example, we know that if you have glue on the leaf, it changes the reflectance. We know if you don't scan it against a black background, it changes the reflectance. We know if you don't have the probe perfectly flat onto the leaf, it changes the reflectance. So this is still very experimental. And some of the things we're trying to figure out is just what matters and what can you ignore safely at different times. The ultimate goal of this is we scan, as many of these plants as we can get through in 6 years. We're starting with trees as a focus because they tend to be longer between flowering periods and thus often hard to identify. And it's just they're important components of tropical ecosystems, a lot of tropical ecosystems. The ultimate goal is that we incorporate those hyperspectral scans with image analysis using machine learning, as well as traits extracted from those images, and build an integrated identification model that you can put whatever data you happen to have available from your specimens into the model, and it will give you a predicted ID. That's the dream. It's been tested in small numbers of groups, and I'll give you a little case study that we did in-house, but it's been done in a few different groups with some really promising results. The big question is what happens when you scale that up and you add a lot of species, and you're not just doing it for a few things, but you're doing it for hundreds of thousands of species. So what we've done so far as an update, here's Matt Austin, my co-PI, and Tyler Hughes, who's one of our hyperspectral scanning assistants, working on a scan. The protocol to do the scanning was finalized with iHerbSpec. I think it's being submitted for publication, but it's available on their website if you're interested. That was important because they had to standardize what metadata they were going to collect. Again, trying to figure out what matters and what doesn't matter. Notes about what the condition of the leaf is, what the temperature was when you were scanning, whether the specimen was, if it was dried in alcohol, for example, or if it was dried at room temperature, or sorry, dried by air, if it was stored in a colder temperature. All those things, we're not sure how they're going to affect the conclusions from the hyperspectral scanning. But they finalized the protocol, finalized the metadata fields, and we've scanned about 10,000 specimens. We're about to have our first 10,000 hyperspectral scanned cake on April 6th, including over 1,000 species from 84 genera of trees. One component of this, going back to that extended specimen network, is we also had to build a module in Tropicos so that we can put those data in and associate them with the specimens. Otherwise, we'll just have them kind of floating in a separate set of spreadsheets. We want the data to be searchable and also to be able to extract trends for different species. So our first pass of this worked on Vitis, which is not really a tree, but we had a preliminary study with the Department of Agriculture to look at Vitis species. And we scanned 11 different species. And this is a confusion matrix. So what this is it has the true identity as according to the label that's on the plant, and then the predicted identity from the model. So all of these species were scanned. Half of them were used to train the model. So you sort of tell the model, here's what the hyperspectral data looks like for this specimen, and here's what species it is. And you feed it a bunch of those until it learns what the expected patterns are. And then you give it a bunch of hyperspectral data from the other half of your specimens that you haven't put in the training and ask it to predict what the species are. So as you can see, the accuracy here is pretty high. On average, it's about 90% accurate, with some species being identified 100% all the time. Where you see these numbers outside, what you ideally want to see is a solid diagonal of dark orange, red 100s. So the higher the number here, the better the model is performing. The lower the number, the worse it's performing. So what's interesting as well is there's some species that seem to be more variable in their hyperspectral fingerprint than others. What's also neat about this is that we can use this potentially as a tool for identifying incorrect species. So the model will tell you with a certain level of precision how confident it is in that particular score. So for each specimen, it'll say, I'm assigning this to Vitis mustangensis with 95% confidence, but it also might be Vitis palmata with 5% confidence. So you can set a threshold and say, all right, I'm going to pass every specimen that has lower than, let's say, 75% confidence in its identification to a specialist. And so what we did is with this preliminary Vitis study, we took all those that had below a particular cutoff of identification confidence. In this case, I think it was 80%, and sent them to Aaron Floden, who's a scientist here at Missouri Botanic Garden and an expert on Vitis. And what he found was that several of them had incorrect determinations on the specimens. So that's why they didn't match what their true identity should be. They were, in fact, misidentified in the herbarium. A couple of them, the original determination was correct, but the model, if you look, wasn't very confident about them. So there was something kind of interesting going on there. And then a couple of them, the characters weren't there in order to correctly identify them. So the names that were on there were done kind of preliminarily anyways. So even if we can't get 100% accuracy, we don't necessarily need that. What we can use this as is a tool to help speed up the curation process for our botanists and help them find specimens that are of interest. Say, here's everything that came from this particular species folder that says it's low confidence as that species. You should take a look at them. Maybe they're misidentified, maybe they're new species, or maybe there's something ecologically interesting. So more recently, Matt Austin ran this again, where he upped the number of species to 18 species of Vitis. So it's the same thing you're seeing before, which is this confusion matrix. You may notice now, if you look at the numbers, they have dropped considerably though. So just adding those, going from 11 to 18, and you have a dramatic reduction in your confidence. Now, part of this is being dragged down by misbehaving species like Vitis vinifera. So this is the common wine grape. I believe there's something like 4,000 or 5,000 varieties reported for Vitis vinifera. So this is a globally cultivated, highly diverse, important crop species. So kind of as expected, it has a very diverse hyperspectral signal, and that makes it hard to do identifications consistently. So maybe things for Vitis vinifera, this is not possibly the best tool, although it's possible that if you knew it was some kind of Vitis vinifera, it could give you interesting information about what variety of Vitis vinifera that is. So it's still, it's reflecting something real that's in the chemistry of the leaf. It's not just making up random numbers, but in this case, it's reflecting the real biology of this is a highly diverse species. We're going to keep on adding specimens. You can also play around with the analytical method that the model uses to identify those. This is kind of a default method that's been used, but that's one of the areas of research we're exploring as well, is how do the conditions around the model affect its performance? And then what else can we do to increase its confidence and accuracy? So besides the core mission, which is that identification, we also have some wonderful postdocs, and they have interesting side projects that they're doing with the hyperspectral data. So this is Riley Fortier, one of our hyperspectral postdocs, and he was interested in a couple different things. One is what the effect of drying method is on the hyperspectral signal. So this is kind of a mechanistic question about do we have to make adjustments for the way the condition of the herbarium specimens? So what he found is that there are significant effects. So where you see the black line spike here, that means the wavelengths in that region are significantly different when you are looking at that particular effect. So in this case, alcohol, you see these two spikes here, drying and alcohol interacting these two areas. Drying is at the top. That's the intensity of drying. If it's very high heat, then it's alcohol, and then the combined effect of the two. So you see a significant effect on the wavelength inference in this sort of just under 2000 nanometer region. What we found is it doesn't seem to fundamentally change our inferences, though. So the identification model and some of the functional trait estimates are what we call robust to that particular factor, but it does show up. So something that we know to keep an eye out for if we're seeing something kind of funny happening, especially in that region. He also looked at leaf mass per area, which you can predict from hyperspectral scans, and found that that was consistent with patterns of climate change, where leaf mass per area is decreasing over time as temperatures and CO2 levels increase. So a sort of secondary validation of hyperspectral scanning as a tool for extracting a very particular ecological trait that normally you would have to measure at, pretty laboriously. Our other postdoc is Ryan Fuller. And so Ryan is interested in polyploidy. And so he's been taking a few different approaches, mostly in rhododendrons so far, for seeing if hyperspectral data can be used to identify different ploidy levels. If you're not familiar with what that is, that just means the number of genomes that are in the plant. So humans are diploid. We get a copy of our genes from our mother and a copy from our father. We're virtually never anything else besides that. When we have different numbers of copies than that, it tends to be related to either termination of the pregnancy or some kind of severe developmental condition. Plants are not that way. Plants can tolerate a dozen copies of their genome. They can duplicate their genomes. And those, what we call polyploids, where they have more than two copies, can really complicate our taxonomic understanding because they'll be different from one another, and they can hybridize often between the ploidy levels. So here's that same confusion matrix, just on a much smaller scale. This is, instead of predicted species, it's predicted ploidy level, so how many genomes. And this is the true ploidy level measured from some other techniques. And what you're seeing is 87%, 94%, 83%, a pretty high accuracy rate for using just a scan with one of these spectroradiometers to tell you what the ploidy level is of those rhododendrons. And then down here is a technique. I won't get into the details of this, but basically you find this higher outlier status of this higher ploidy level compared to the diploids. So here's the tetraploid that has 4 copies in light blue. In brown is triploid that has 3 copies. And then in dark blue is a diploid, with one exception here, which is supposed to be a diploid, but there's something funny going on there that they're going to look into. So another way that we can use this technique to infer interesting things about the biology of the plants. So that third component then, the curatorial mentorship program, all of those models that I just showed you rely on training data. And the training data, so your model is only as good as your training data are. If you have inaccuracies, major inaccuracies in your training data, your model is going to give you wrong predictions. So it's critical that we have, to start, as good of names as we can get on the specimens that we're using to train these machine learning models. A lot of our scanning staff are coming to us with sometimes undergraduate coursework in botany, very rarely a little bit of experience working in herbarium as an undergrad, although that's a pretty rare case. In some cases, no botanical training at all. We have some anthropology majors, I forget some of the other majors, an engineering major. So the mentorship program is about a quarter of the digitization staff's time. So all the supervisors and scanners that I showed you on that org chart slide that had all their lovely faces are participating in this mentorship program. And they do workshops. Like here, there was a workshop where they were looking at type specimens. Here's a workshop where they're, these are all workshops, learning basic plant morphology. Here they're with Julian Aguirre Santoro looking at ideas of field collecting and identifying epiphytes in our climatron, our tropical glasshouse. And then here with one of the horticulture staff looking at arid plants in our arid house. And then they're also paired with a curator mentor on an individual project, curating some part of the collection. So identifying specimens, preparing the data for red list assessments, updating taxonomic changes. So if there's a publication that comes out in a group and says, oh, here all these names need to change, they'll make those name changes. And just learning from that curator, how do you go about taking a group of plants in the herbarium and making sure that the names are as accurate and up to date as possible. And I just have a couple uh, examples. So these are two of our, well, Gabrielle is our hyperspectral scanning supervisor. Wilson is one of our our herbarium digitization assistants. We have HDAs and HSAs, herbarium digitization assistant and hyperspectral scanning assistants. So Wilson is working on the Morning Glory family with Doug Stevens. So he's identifying some species, some specimens, and preparing a key for the genus Evolvulus for Southeast Mexico. Pretty sure he discovered two new species as part of this process that were just in the collection and hadn't been discovered or written up yet. And Gabrielle is working on the tea family for the Cambodia, Laos, Vietnam region with Carmen Puglisi, and started out compiling a checklist of the species in the family, doing literature searches, compiling data from GBIF and from our herbarium. So it's been the third part is by far my favorite part of the project. The hyperspectral scanning is obviously really cool and science fiction-y. It'll be wonderful to have the herbarium digitized, but this is such a rare skill set that's really hard to come by training opportunities for, that it's been really gratifying to see how much this cohort of people who have mostly, other than one who left for a permanent position with us in a different area. That's actually Tabitha right there. She's now our shipping assistant. Um, but they move kind of as a cohort together, and really seeing their skills develop has been tremendously gratifying and an interesting challenge of how do you train someone to do this. Many of even the professional botanists kind of learned to do these things on the fly. They didn't have formal training in it. It was as part of a project they were working on, or they had a particular mentor who taught them. So that is a broad summary of a pretty quick summary of what we're working on with this project. We're about in year 2 of the project, so a little over 4 years left. We just recently, well, actually, just recently we hit 1 million specimen images, but I don't have a picture for that yet because the celebration is on the 6th. But this was from our 500,000 celebration. I promised them cake every 500,000 images and every 10,000 hyperspectral scans. So if you're interested in these data, keep an eye on Tropicos. They're going on there at a pretty stunning and alarming rate. And there'll be publications coming out soon, I think, or submitted soon about the hyperspectral research. And if you're interested, we'd be happy to hear from you and talk about collaboration possibilities. It's a highly collaborative project. A lot of the stuff with the hyperspectral scanning is very new. So always looking for new ideas. And I'm happy to answer any questions.
At this point, if you have a question for Dr. Teisher, please unmute and ask away.
Okay, there you go.
Paul Catling, I have a quick question about the improvement of the identifications with increasing numbers of species of Vitis. And that is, I'm wondering if one included the hybrids and the infrataxa, one could make it more accurate. As it stands, it seems this methodology could include the difficulties presented by hybridization, but it hasn't yet done so.
Yeah, absolutely. That's a great suggestion, and you're correct. So this was still a very preliminary pass of kind of just the bare bones model of just putting the hyperspectral data in and nothing else. But as you mentioned, there are a lot of different supplemental information, supplemental data that you could feed into the model to help it tease apart those patterns better. Another one would be geography. So we didn't tell the model where those specimens came from. We just gave it a hyperspectral signal and a name and said, how are you doing with that? But if that becomes, for very widespread things, they're going to have a tremendous amount of variation across their range. One is to incorporate subspecific classifications if you have them. Another is we could just also feed it the country and say, all right, here are the patterns for this species from Brazil. Here are the patterns for this species from Colombia, and see how it does with that kind of information. But yeah, great point.
Other questions? There's one in the chat. The question is, does the age of a specimen, i.e., how long the specimen has been mounted on the sheet, and the density of the trichomes um, affect the reflectance data?
Density of the trichomes, almost certainly, because that will be a physical effect of the reflectance. I'm not positive about the age of the specimen. I have some vague recollection that someone was looking at it, but I don't know, not in our group, and I'm not positive if they're at a point to share results yet. It seems to be fairly robust to it, but there's a lot of those basic questions of, if you have a species, for example, that's highly variable in how hairy the leaf is, uh, that could really throw a wrench into that particular species. Something is highly variable about Vitis vinifera, for example. I don't know what it is that's causing the tremendous range of those scans. There will be, we anticipate, groups for which this works really well, and there already have been, a few test cases, and there will be groups for which this works not so well. And so one of the interesting and fun things is just it does tell you something about the leaf that you're scanning. And so figuring out when it can be applied, when you have to incorporate some of the supplemental data, when you're probably better off using a different technique because that particular group is just not really amenable. For example, unfortunately, I don't know if I have an example. The probe area is about the size of the bottom of a pen. So it's not a pinpoint scan. So if you have something that has really, really fine leaves, like some conifers with their needles, it is not easy to get a clean scan from this. That may change in the future. We're also hopeful that you can do hyperspectral imaging, for example, from a drone or from some satellite data, and that if we can build these profiles, that we can expand the number of applications. But starting with herbarium specimens, because they tend to be the most accurately identified. Two, you can always go back to them and check the identification if something turns out to be weird. Three, because they're dried already, and the preliminary results that some people in this field have Sean Kothari, that's the name I'm trying to think of. Sean Kothari published a paper with some colleagues a few years ago, finding that dried leaves perform better than fresh leaves because it removes the effect of water, which is variable within the plant anyways. So herbarium specimens have a lot going for it to start out as kind of building a library for this process.
There's another question in the chat, which is how can people viewing tonight's program contact you about the orchids that have been studied?
My email is jteisher@mobot.org. I don't know if there's a way can I post in the chat?
Yes, you can.
I will try that. You are welcome to email me, and if I can't answer it, I will direct it to somebody who can.
I have a question myself. I'll get that in, and then I'll pick another question from the chat, which is for the oldest orchid specimens in particular. I mean, as someone who, has read a lot of articles about the early plant collectors for cattleyas. They were fairly coy, if not disingenuous, about the locations where the plants were collected. Is that a problem, or is it not a problem because what was collected never made it to the herbariums in the first place?
That's a good question. I mean, it's a problem if you're it depends on the application, right? So if you're trying to understand species limits and you have the specimens and let's say that they fudged the locality by a few dozen miles. You can still probably wrap your head around the patterns of variation that are in that particular species, and it could still be a useful specimen. Similarly, if you're doing phylogenetics and you're just trying to construct an evolutionary tree, it can still serve as a useful exemplar for that purpose. If you're trying to understand ecology, especially adaptations to microhabitats, for example, well then, even if you're off by a few feet, it can be significantly different if you were in a waterfall versus on a tree. That's a fundamentally different kind of mini habitat. So yeah, I think it just kind of depends on the application. Similarly, coordinates are not all created equal. If you look at an aggregator like GBIF and you map all the specimens, they will map a point on the map, but that doesn't necessarily mean that that's the level of accuracy or precision of, or the level of precision of that coordinate. It could just be what it gave you was only, you know, one decimal place and it had to put it somewhere, so it put the dot there. But in reality, your error radius for that coordinate is massive. So older coordinates from specimens will have these massive error radii compared to a modern coordinate that can be quite precise. So it's kind of up to the researcher to figure out what data can I actually use for the question I'm trying to ask, and what specimens I'm going to have to ignore. Does that answer your question?
It does. Thank you. Here's the question from the chat. What about species that are presently distinguished by their roots?
Um, what do you mean? Like,
I'll turn it over to Cindy. This was her question. Cindy, if you can you embellish your question?
Yes, I believe species like some of the delphinium require a plant to be dug up, and based on the roots, they can distinguish some different species. So I'm wondering if you've had any experience looking at the multispectral signature of those to see if it can be used in lieu of digging up a plant. I think that would be great.
yeah, gotcha. Yeah, sorry. That makes perfect sense. We've not yet, but one kind of comparable area is that we have an enormous number of sterile specimens in the herbarium. So, especially ecological monitoring plots, you kind of just have to collect when you're there doing the monitoring. So you maybe draw a transect if that's the method that you're using, and you just collect everything that's above a certain size, diameter at breast height, trees, for example. So whether they're in flower or not, and all those specimens come back to the herbarium. And we have curators who look at these and just say, I don't know what you want me to do with these. You know, these are hard to identify when they're in flower, and they're impossible to identify when they're not. So similarly, if you had those orchids without their roots, those specimens would be really, really tough because you don't have the key features needed to identify them. But it's an ongoing question of can presumably they do have different chemical compositions in the leaves. It's just not something that we can see. So that is an area where we're especially excited of those species that are hard to identify from herbarium specimens, often don't have the key features, but may be amenable to hyperspectral scanning. So one family is the Lauraceae, the laurel family. Really hard to identify if you don't have flowers. From what I understand, hard to identify even if you do have flowers. And we have a lot of sterile specimens of them. So that's one of the families that we are testing this method on to see if it's effective.
Any other questions from the group? If not, then I would like to wrap things up and thank everyone for attending tonight. Um, just a reminder that this program has been recorded and we'll be posting it on the NOC website very shortly and also on YouTube, something we're trying for the first time. This is also the final program of the NOC's spring series of online programs. Our new programs will begin in September, and we hope to see you then. So, again, thank you. Thank you, Jordan, for tonight's program, and hope to see everyone in the fall. Good night.
Thank you.